Perfect. Thank you so much. So this talk will be recorded, so just so everyone knows. Dr. Gepts has also agreed to stay after to speak with graduate students about his career trajectory, his work, or anything else that comes up. So please hang around for that if you would like to. So with all that said, I am really pleased to introduce Paul Gepts, a native of Belgium and distinguished professor in plant science at the University of California, Davis. He has developed an international research and education program with activities on 4 continents, including Europe, the Americas, and Africa. His research is focused on the evolution, genetic conservation, and utilization and breeding of crop plants. He has emphasized Phaseolus beans because they provide an excellent experimental model to study the process of domestication, including a phylogeny of 7 domestications in the genus, the geographic and ecological distribution of these domestications based on field explorations, the genetic and molecular basis of the domestication syndrome, the co-evolution between bean hosts and their pathogens, and the development of a domesticated Phaseolus pangenome. His diverse research group at UC Davis has hosted students and scientists from all continents. He's the recipient of the Crop Science Society of America International Crop Science Award and the Agropolis Louis Malassis, I hope I said that right, I probably didn't, Prize, and Fellow of the American Association for the Advancement of Science, the American Society of Agronomy, and the Crop Science Society of America. He has published some 250 articles or chapters and released several common bean and garbanzo varieties. So I will now pass it over to Paul and we can give him our undivided attention.
So thank you very much, Bailey, and thanks also to the department and Eric in particular for inviting me. Can you hear me loud and clear? I want to make sure. Is that okay?
Yep.
Yes, it's great.
Okay. Yeah, I'm actually from home here, as you will see. And I would like to thank you. It's a real pleasure to give this seminar. Before I get into the subject, I would like to acknowledge the students and postdocs as well as the funding sources. What you see here to the left, this is how we meet these days. It's often by Zoom, and so although depending on the prevailing variants, we can also meet outside and appropriately distanced. Currently, we are meeting by Zoom, but I think that starting in April, we'll be able to go back to a kind of a more in-person, but still distanced outside meetings. Now, the work I will talk about primarily is recent work, although it builds on, I would say, several years, if not decades, of research on the domestication and particularly of Phaseolus beans, and you'll understand why that is in a few slides. So our work is primarily today is the work of Travis Parker, who is a postdoc, and this weekend became a dad of a first child, actually. So, and then also of Dr. Sassoum Lo, who is also a postdoc, and the two have been working together on the development of pods in beans. And as Bailey said, I work internationally, and the most recent research is really collaboration between people not only at UC Davis, Travis and Sassoum, but also in other institutions in the United States, in Mexico, Colombia, Brazil, and also in Africa, in Kenya. And I find that very satisfying to bring together people, even during this pandemic. Sarah Njau is a student from Kenya, and she's spending several months at UC Davis. So domestication really represents what I would say a fundamental change in the relationship between humans and the natural world. And for millions of years, humans have been hunters and gatherers. And so some 10,000 years ago, for reasons that we are still exploring, there was a change. And so domestication really is a selection process for adaptation to cultivation and consumption conditions imposed by humans. And it does represent a major change because instead of being hunters and gatherers and really being the recipients of the natural riches provided by our planet, humans became active exploiters by the act of planting to supplement and even to overtake the provision of these riches on our planet. And so it's really a change in mentality. And people would argue that starting 10,000 years ago, over that short period of time, we have become exploiters and even over-exploiters of the natural world provided by the planet. Now, I just said that we don't know why that change occurred. It's an active field of investigation. There is a group of people who have tried to establish a single paradigm, at least for grain crops, as to how domestication took place. Their fundamental finding, which can be disputed, is that domestication was slow and gradual and took place over several thousands of years. In order to account for that and to support that, they argue that it was geographically diffuse and a repeatedly recurring event. And also, in order to account for the slowness, is that there was no human agency in this process, or very limited, or not until later. Others, however, argue that there are probably multiple paradigms, that it could have happened much faster. The argument in favor of this is that, as I will show here in genes, is that you have major genes involved that are highly heritable, and so progress from this in this selection process that represent domestication could have been very fast, and you can model it that way, and also that cultivation of undomesticated plants in most cases would not have been very rewarding. And the experiments of wild pea planting of Shahal Abo and his collaborators in Israel is an example of that, where you get negative. Actually, you plant more than you actually harvest. Arguments to the contrary of this is the mutation rate. That's the limiting factor. And also where these mutations can be found, whether it's part of the standing variation or a de novo mutation. Now, the same multiple paradigm argues that it's a localized event, and the data, the molecular data, seem to suggest that, whether it's a single or multiple domestications, you can identify regions where you have a close relationship between wild and domesticated types. The caveat here is that even in selfing species, you have gene flow between wild and domesticated types, and that confounds this identification of original domestication factors. But with genomics, we can actually get around this confounding effect. The other aspect is that it's hard to imagine, I think, that domestication and management of plants would have been unintentional. I think that there was probably an intense knowledge of plants that have been domesticated in many cultures, even among hunter-gatherer groups. There is evidence for plant population management as a prelude to actual domestication by planting and so on. And there is also evidence of pre-domestication detoxification, for example. And you have to ask yourself, why would people domesticate a plant if they could not use it as a food and so on, because they had not learned how to detoxify it? So there is this accumulation of knowledge that have preceded domestication. Anyway, that's a kind of a general framework against which we do our research. Now, there are multiple reasons to study domestication. I've told you it is a fundamental episode in human evolution. For millions of years, we were hunter-gatherers, and then about 10,000 years ago, which is fairly recent, we switched to a more, you could say, proactive phase of our evolution in terms of exploitation of natural resources. Now, it is also a model for evolutionary studies, and Darwin realized this in 1859. He published The Origin of Species, and in that book, the first chapter is devoted to variation of plants and animals. And he was faced with a conundrum. He argued that species are not static, they evolve, and the engine of the evolution is selection for fitness. Now, selection for fitness would be way more efficient. It was heritable. The effects of that selection were heritable. 1859, it's a time, it's before Mendel, and it's before of many of the genetic research in the 19th century that identified chromosomes, meiosis, mitosis, and so on. So he was able to demonstrate the effects of selection with plants and animals. He was himself what is called at that time a fancier of pigeons. He was raising pigeons, but you could do the same type of observation. And within a decade, he had published an additional volume titled Variation of Animals and Plants Under Domestication. So he demonstrated within the lifetime, and I would argue within the time span of a PhD thesis research, that yes, you can see the effect of selection. Okay, and so natural selection could really be the engine of evolution, and you can identify the model here has advantages because you can identify progenitors. The time frame is known more or less through archaeological data. You have targets of selection, and we'll talk about this domestication syndrome. And in these species, in crop species, or you have genetic and genomic resources that are more abundant. So the third major reason is that this domestication process is a key to understanding the organization of genetic diversity and adaptation of a crop. Now, you can take domestication, you have various definitions, but I would argue that domestication actually starts way before the actually cultivation of a plant. There are events before the actual domestication about 10,000 years ago that are equally important, if not more important, than the domestication process itself. I've listed here the pre-domestication divergence and adaptation in the wild progenitor and the related taxa. Also, this process of awareness, knowledge accumulation, and gathering of the plant must have taught a lot of, or must have given a lot of information to these hunter-gatherers. And so eventually you get to this pre-domestication cultivation, which I think is a necessary condition for domestication, the actual domestication process, and then subsequently, which is equally influential in terms of the genetic diversity and so on, is the dispersal from the centers of domestication. Some crops, it's really a worldwide dispersal, and further selection for local adaptation uses and plant breeding. So this is a fairly expansive definition of domestication. You can have a more narrow and the narrow domestication in the narrow sense would have occurred about 10,000, 12,000 years ago, and that would represent what we call domestication in the narrow sense. Now, why is this of interest? It's because of genetic conservation and the use in plant breeding. This is kind of the plant breeding pyramid, where most of the plant breeding takes place with advanced cultivars. But in order to accomplish some of the goals of plant breeding, you need to descend into the land races or even the wild types to find the necessary disease resistances or stress tolerances. Okay. So based on this research on domestication, you can identify spatial patterns. Where in the world is the genetic diversity of a crop located? Even the temporal patterns, the sources of genetic diversity, which populations, and so on. Where do you find adaptation to stresses? And also identify novel germplasm sources. So there is an applied component in addition to these more fundamental components focused on the understanding of human evolution as well as the process of evolution itself. Now, the domestication process itself is fascinating. It relies on factors that related to the plant or the animal, if I were to give a talk on animal domestication, also the environment. And what is fascinating about domestication itself is that there is, of course, a human component. You could say that these factors here to the top and the right are characteristic of natural evolution, but in crop evolution you add this human component, not only 10,000 years ago, but also currently, because farmers, especially in the centers of origin, manage their crops and their seed stocks in such a way that it has an effect, and domestication is a continuing process. And the seeds or the planting materials in these types of agriculture, in these agroecosystems, becomes part of the, you could say, the living capital of the farms, unlike seeds in an industrial type of agriculture. So why the genus Phaseolus then? It turns out that it is a domestication hotspot. And you have 5 domesticated species out of about 70 to 80 total species in the genus. And you have 7 domestications because 2 species were domesticated twice. Now, I will give you a quick overview of these 5 domesticated species. You see here the distribution of the wild progenitor. And it goes from the most widely distributed species, common bean, then lima bean, and both have a distribution not only in Mesoamerica where the genus originated, but then extending into Central America and even into the Andes all the way to northwest Argentina. And you see the same thing, but to a slightly lesser extent for lima bean. These are different species. Then you have runner bean, peppery bean, which then is limited to northwest Mexico and the southwest of the United States. And then the year bean, which is probably the least known domesticated species, which originated in a few populations in the highlands of Guatemala. Okay. And so through these molecular analysis, we are closer to identifying the actual area of domestication of molecular variation in both the wild and the domesticated types. And so you see most of the domestications took place in various places in Mexico that represent different ecologies. And you have then 2 additional domestications in South America that resulted from rare long distance dispersal events. You have one here for lima bean in the lowlands of Ecuador on the western side of the Andes, and another long distance dispersal here in the southern Andes, probably between Bolivia and northwest Argentina, for common bean. So common bean and lima bean both were domesticated twice in Mesoamerica and in the Andes. So here you see the 7 domestications. You have the top row is the seeds of the wild types, and then the next row is the tremendous diversity in seed types that exists in each of these species and were selected by humans since domestication, the original domestication. Now, how do these species differ? They are truly different biological species, although most of them can be crossed, being lima bean. They have different adaptations, and so the question is, why did farmers then select different species? And there seemed to be an alternative. You could say you can select one species and then adapt it to different ecologies, different environments, or you could domesticate several species, each with its own adaptation. And that's apparently the model that has been followed in Phaseolus. So you have, actually they are listed here, you have runner bean and year bean are mostly in cool environments. And farmers know that, actually, in Mexico, for example. You have common bean, which is a kind of intermediate environment, not too hot, not too cold, not too wet, not too dry. You have then lima bean, which is probably the species, even before domestication, that has the broadest adaptation. And then peppery bean, which thrives in hot and dry environments in the northwest of Mexico, southwest of the United States. They also differ in terms of the range of growth habits, depending on the intensity of domestication and also on their reproductive system. Okay? So one of the questions is also, in addition to why have 5 domesticated species, why is then common bean the most widespread and most economically most important species? And we don't really know that for sure, but this is a question that is now coming up. I can't answer the question. I can only speculate on that. So common bean is the species that has been studied the most because of its economic importance. And what you see here, this slide, then gives you a kind of of evolution. You have originally an evolutionary lineage in Mexico, what is now Mexico, that existed for at least 500,000 years. And it's a long distance rare dispersal event distributed wild populations to Ecuador and northern Peru. The location where you have the Andean lima bean domestication, but at higher altitudes. And because of its differences, it has been kind of renamed as a separate species. Then a second dispersal event to the Andes of the southern Andes, so southern Peru, Bolivia, and Argentina, gave rise to a wild southern Andean gene pool, and eventually then you also generated a wild Mexican gene pool, and domestication took place in these two lineages in Mexican Mesoamerican gene pool and in the Andean gene pool, the blue dots here. This, the yellow one here, the intermediate one, was not domesticated. And so our understanding now of genetic diversity is in common being relies on this scheme, and I'll use it later on in my talk to point out some of the diversification along these different evolutionary lineages. So in my talk, I'll come back to this domestication triangle, which has informed our research as well as our teaching, actually, because I teach a crop evolution course every quarter at UC Davis. And I'll deal with this aspect of the interaction between plants and humans, but eventually also I'll bring in the environmental effect. So the domestication syndrome in common bean has been defined in the, I would say, 80s and 90s. We had a paper out by Koinangi et al. In 1996, which was an RFP, the genetic control of this domestication syndrome. And to me, it is very heartening that the predictions we made as to the locations of these for the domestication syndrome is now largely being verified, because we have been able, and I say we, the Phaseolus community, has been able to identify the genes that underlie these QTLs identified in the 1990s in that 96 paper. So these are different elements of the domestication syndrome. I will talk mostly about the an element in the change of the growth habit, the determinacy trait, and the pod shattering and pod string. But I've already alluded to this tremendous diversity in the seed types, which is the harvested organ, the dry beans, but also among the pods for the green bean types in common bean. And also there is variation for time to flower, for photoperiod sensitivity, and seed dormancy. Okay. So I'll deal mostly in the first phase with the determinacy issue, and then later on I'll talk about the pod traits. So you see here, this is a photo I took in Mexico, and it is somewhat difficult to see, but you see the leaves here at the bottom. They represent a teosinte plant, which is a wild maize plant, and at the top you see here, these are the pods of a dry senesced wild bean plant. And in a sense, it's a preview of what people were, the ecosystem that people would develop, which combines, again, maize, domesticated maize now, domesticated beans, as well as domesticated squash, and that is the Three Sisters or the milpa system. And eventually in beans, they developed not a climbing type, but a bush type, which is a determinate bush. Not all bush types are determinate, but in this case, they are determinate. And so this determinacy means really a limitation in the growth of the number of units, and a unit being a leaf, an internode, and an inflorescence. And so plants grow by this in modular fashion, and the gene is important because it limits this number of modules that can be piled up, actually. An additional reason, or an additional correlated trait is the earliness in these determinate types. And you find the determinacy in common bean as well as in lima bean. And for example, in California, they grow both viney types, which is really a climbing bean, and a bush type, which is a determinate type. Now, we took a candidate gene approach by taking Arabidopsis genes and determining which ones would maybe co-segregate with the determinacy trait in common bean. And it turned out that there was actually a mutant in Arabidopsis, TFL1, which co-segregated with a determinacy phenotype in common bean. We extended that from a small population of about 100 to a much larger population, still co-segregated. We did quantitative PCR. This particular gene did not, had very low expression, consistent with the recessive nature of determinacy. And eventually, Shelby Repinski, now Shelby Ellison, did the transformation of Arabidopsis with a functional determinacy allele with common bean, and she was able to restore the phenotype. So here you have the wild type Arabidopsis, you have the TFL1 mutant in Arabidopsis, and then this is the transformed Arabidopsis with a functional indeterminate sequence from common bean. And so you see that it restores the wild type phenotype. And so this was a strong evidence that, yes, we had identified the particular gene. Myung-Hae Kwak was a student at that time from South Korea, and she then proceeded to look at the variation for this particular TFL1 homologue in common bean. You see here the gene itself, PVTFL1Y. It has 4 exons, 3 introns. Among the indeterminate types, she looked like, yes, there is sequence variation, but none that would invalidate or eliminate the function of the gene. So she only found mutations that represented loss of functions in the determinate types. Surprising to us was that it was not just one mutation. There were like 6 or 7 mutations, and they represented the variation you can expect. You have frame shift mutation, you have deletion of an entire locus, you have an exon intron splicing failure, for example, non-synonymous substitution, and then the most important one, the most frequent one, was a retrotransposon insertion of about 4,000 base pairs into the 4th and last exon. And so she had to sequence, in order to have a complete picture, she had to sequence, do extended sequencing into these 4,000 base pair region, and she then ended up in the next gene. Now the question is, why is this retrotransposon the most frequent one? It's almost the exclusive one outside of the Americas, for example, in Europe and Africa. We don't know for sure, but our hypothesis is that it's related to the gene content outside the PVTFL1 locus. And so these linked traits also influence the phenotype. And that is probably the preferred one, was the retrotransposon insertion. And we have done the back crosses and look at these results, but we still have to analyze them. So determinacy has multiple origins. I've listed a few of them. Most of them are Andean. There is one here that represents an exon in transplicing failure. It's American. So it's a typically Andean trait, the Andean domestication. So let me then go over the second aspect of the domestication, that is pod shattering and also pod string. So this is an evolution. You see here the evolution from wild to dry bean. And in common bean, you have an additional commercial class. It's the green or snap beans. All of them belong to the same species, but you have two phases. You have this reduction in pod shattering, and then you have an elimination of the strings. It used to be called that green beans used to be called string beans, but they are now snap beans or stringless beans. And the genetics of this is epistatic on the genetics of the first phase. Okay, and so, but what are these mechanisms? So here you see a wild bean plant, and the green is something else. It's not beans. The plant is mature, it's dried, and it still carries for a few weeks its closed pods. But eventually, the pods are going to open, and the plant is going to distribute its seeds. Now, this is an explosive mechanism. And so people talk scientifically about pod dehiscence, but in everyday language, we talk about pod shattering. And it distributes the seeds in a circle about a radius of about up to 5 meters. Okay. And if you take a close look here, for example, what you see is that the pod opens, it has two valves, they separate, and at the same time, you can see that the two pod walls are strongly twisted, tightly twisted. It's that it creates a slingshot movement that so that the seeds don't drop vertically to the ground, but also are distributed in this circle of about 5 meters. Okay. Now, so what is the mechanism? Now, this is very important because the pod opening, the shattering, needs to happen when the pods are dry. And so if you look at the distribution of these Phaseolus, domesticated Phaseolus species and the wild species, there has to be a dry period. Okay, and you see that, for example, I did a principal component analysis of the environments in which wild beans grow, and all of them have a dry period. Sometimes it's very short, 1 to 2 months. Sometimes it extends to 6 to 7 months. And the domestication happened actually was at the extremes of the distribution, where you see the longest dry period in their cycle. If you don't have a dry period, you see this is what happens when you have germination in the pod with a complete loss of fitness. So what is this pod shattering? It relies on two mechanisms. One of them is the presence of wall fibers that line the inside of the pod and that have fibers, lignin fibers, that are oriented obliquely, not straight longitudinally, not transversely, but obliquely, and that causes the twist you see in the pod walls. The other mechanism then is that there is a dehiscence zone here at a suture, and this is the, I think it's the ventral, I think, suture here, but you have the same on the dorsal suture, and this dehiscence zone is part of that mechanism. So I think that the fibers in the pod wall here, they shrink and they create a tension that will then lead to a separation of the two valves along the sutures in the dehiscence zone. Now, when you compare wild beans and dry beans, the main morphological change you see is the thickness and the amount of fibers in the pod walls, inside the pod walls. Okay, we don't see that much change in the dehiscence zone. So we did a study in the genetics of pod shadowing. We used two approaches, use a biparental cross, and then also Anna Ladrida-G was study combining two panels, corresponding to the two domestication, a Mesoamerican diversity panel and an Andean diversity panel. And the two approaches give us the same result with a major QTL on the chromosome 3 by different methods. I won't go into that, but the more sophisticated the method, the higher the lot score, which of course is very good because we keep confirming that same location. The least sophisticated method was to say shadowing or no shadowing, which you can do in the field, and that still gave us the same location. Now you also see that there are other locations, other potential QTLs here, whether in the Mesoamerican diversity panel or the Andean diversity panel. And those will be important for the discussion. So this QTL on chromosome 3 appears to be unequally distributed in the Mesoamerican domesticated gene pool. Its frequency is very abundant in what is called the race Durango, which is a race of northern Mexico, a highland, a dry climate. It's much less frequent in race Mesoamerica, which is a lowland race, moist environment. And you see here, so this is the first axis is a principal component axis of molecular diversity, and you see the contrast between highland dry and lowland wet here, and then the proportion of dehiscent pods, so the amount of shadowing. You see that this allele here on chromosome 3 causes a sharp reduction in the dehiscence. And it's almost all of the materials of this race have that allele. And our interpretation is that in order to migrate into this much drier area, it had to acquire a resistance to pod shadowing. Okay? So conversely, resistance to pod shadowing is also an adaptation trait to drought. There is a candidate gene. It's what is called a dirigent protein. It's involved in polymerizing lignin subunits. Okay? It's a simple genetic control, and so it has been used in breeding, for example, by Steve Stempel, who's a colleague in the department, to develop varieties that are resistant, that are tolerant, uh, resistant to the pod shattering. Okay. Now, the interesting observation as well is that even though the resistance to pod shattering is a domestication trait, it subsists in certain domesticated classes of common bean, particularly in the cranberry types. You see here the percentage of pod shattering can be quite high, cranberry and kidney beans, but also, for example, in small black beans. And so it is very low in the Res Durango, for example, for the pinto beans and so on. So why is that, that a trait that is deleterious in a sense seems to subsist? And we suggest that there are multiple genes for resistance, pod shattering tolerance. And so they can complement each other, even though the parents are resistant to shattering. The hybrid in the hybrid, you can have a complementation, and the shattering, pod shattering reappears. And if you don't have the environment, the dry environment to reveal pod shattering, it passes through the selection of a breeding program. And so the pod shattering gets dragged along into the selection process. And so in California, we are in a good position to detect pod shattering because we have very dry summers. Okay. And so our hypothesis here is that domestication took place in central Mexico, probably here in Jalisco, Guanajuato, and subsequent to domestication, you had differentiation of these races, Res Durango, higher aridity, and selection for these to pod shattering. On the other hand, you had differentiation into Res Mesoamerica, which is a lowland, more humid race, and where tolerance to pod shattering was not necessary nor selected in this case. Okay. Now, similar observations had been made in soybean, for example, in the gradient in Eastern Asia, as well as in cowpea, that is the work of Sassoon, specifically where you see gradients, north-south gradients in terms of humidity, and you see that the tolerance to shattering is present mostly in the northern parts of the Sahel, compared to the areas that are more humid closer to the Atlantic Ocean. So let me illustrate. So you have a major QTL on chromosome 3, but you have these other minor factors that are shattering resistant. In the Andean gene pool, you have a major QTL on chromosome 5 and others as well. So when you cross them, you can complement actually these genes, and you will recover, so to speak, the shattering trait. Really. So let me these are the last slides. I wanted to then talk about this pod string, and the absence of pod strings. It used to be called, green beans used to be called string beans. Now they are stringless beans. And that is because of a mutation that was selected in the 19th century that eliminated these fibers or strings in the sutures. So what you see here is a more detailed analysis. You see a cross-section of a pod. You have this dehiscent zone here, is a structure called the bundle sheath that covers the bundles of xylem and phloem, and you have the linear parchment layer inside the cavity of the pods. It also contains fibers, and so this is a micrograph after staining for cellulose. It's a fluorescence microscopy. In the shadowing suture here, this part here, you see that there is a dehiscence zone here where the cells don't have as much secondary wall buildup. And so when the fibers in the pod wall create this tension to separate, create this tension when they shorten, when they shrivel, it generates this tension, and this dehiscence zone is thought to be a zone of weakness compared to the rest of the bundle sheath cells. And that's where then the split happens. If you harvest the green beans earlier, you're still left with these structures here that are to represent the fiber or the string that people had. In a non-shadowing suture, you see that there is no zone of weakness here. All the cells have this secondary wall buildup, and so the pods, these green bean pods, do not shatter or open at maturity. And the corollary that is that earlier in development you don't have fibers in these sutures, no strings. Another feature, negative feature of this mutation is that it's unstable. So it back mutates to the stringy type. And all bean varieties have that feature. It is a big problem for the seed industry because they have to actively rogue the seed production fields for these revertants. And they have to send people down the roads to eliminate these revertants. For geneticists, this is a dream come true because we have a kind of a isogenic line. You have the original stringless and the revertant, which is stringy. And you can compare them, and then it facilitates pinpointing the genes responsible. So the stringless mutant was identified in the 19th century by Calvin Keeney. No strings is dominant. So this is a mutation that went from a state from recessive to dominant, which is an exception. All these other mutations of the domestication syndrome so far were recessive. This one is dominant, and this correlates, you'll see, with increased expression. Tanya Gioia, a student from Italy that's working in my lab, identified or determined that the stringy mutant was located near a homologue of Arabidopsis, dehiscent. Haggerty in Jim Myers's group at the Oregon State identified a 500 kb region that included PV int, and we have these revertants that we worked with. You have 7 revertants here, or 6 in this case, and we took one of them to have a whole genome sequence. And in particular, we wanted to go after that 500 kb region here and see what the sequence was compared to other whole genome sequences that were available. The result of this was that in dry beans, where pod strings are present, and this is one of the whole genome sequences, you have one copy of this PV int. However, in high style, which is a snap bean variety, there is a duplication of that gene as well as a retrotransposon insertion. And we think right now that it is the retrotransposon which activates the overexpression of PVN. These are the current status of affairs. We'll need additional research to confirm this. Okay, so you see here the correlation between retrotransposon duplication, the absence or the presence, as well as the string score. When it's absent, you have a lot of strings. When it's present, you see no strings. And you see also the differentiation here in blue, the expression of the gene, which is significantly higher than the expression in the revertants, so these isogenic lines. So the reversion somehow eliminates the overexpression of PVA. This then provides kind of a direction where we have to go if we want to solve this problem and achieve a stable snap bean stringless expression. So the conclusions then is that we have multiple domestications. People say, well, this is a replication. I don't quite agree with that, but at least it provides us with a lot of materials and for comparative genomics. It's important to understand plant and crop function to know these domestication traits. Domestication introduced a radically different selective regimes, and we see that what was true, a plant that operated sometimes for millions of years, with the arrival of domestication, the selection regime changed completely. And what was deleterious before domestication became a functional advantage after domestication. So you see these contrasting selective regimes. The diversity of domesticated species also provide the opportunity to breed for climate change adaptation. And I think particularly of lima bean, but especially of tepary bean from the northwest of Mexico as providing adaptation to heat and drought. Domestications we see are most often loss of function, but there is this important exception, the loss of strings, which is dominant. There is a phenotypic conversions, but the molecular conversion is less obvious. So different genes may be involved in phenotype. The genetic redundancy then that follows from that, or the multiple allelism, leads to reflects the need to have both GWAS and biparental or multiparental crosses, and it's also a cause of the persistence of some of these pre-domestication or ancestral traits. In this particular talk, I've used approaches of synteny, comparison side by side in these isogenic lines, phylogenetic, genealogical, ecological mapping to understand these patterns of diversity imposed by the domestication process. I've generally found this the study of domestication to be a fascinating area, actually, and maybe personally it satisfied my interest in history, and evolution is the history of biology, also geography. So we continue to do that, and I've found it extremely interesting. And I thank you for the opportunity to give this presentation. Thank you very much.
Thank you so much, Paul, for clapping here. So we have time for a few questions. We're a little over time, so I do want to be respectful of people's times, but there is one question from Yolanda in the chat that I can ask now. She had to go, but I'll fill her in later. She says, I'm interested in the geography of bean domestication. I was wondering if you have a paper on the geography of bean domestication. I am wondering which populations in Jalisco appear to be most likely founders.
Yes. So we have a paper. There is a paper. Let me by Kwak, Kwak, that was published. Let me check here.
You're also welcome to stop sharing your screen if that's easier for you.
The it was published in Crop Science. I'm hesitating about the year. It's 2009. I can put the in the chat. I put the reference in the chat, and it says, where was it domesticated? In the Mesoamerican domestication. The Andean domestication would be a paper by Roberto Papa of Italy, who was a former postdoc in my lab. And the Quack paper tells you the specific areas or the specific populations that are the closest to the domesticated gene pool. And it's really interesting because it's a zone, a kind of east-west zone in between two mountain chains, the central volcanic axis and the west Sierra Madre, and it extends west-east. And actually, we visited that area, and the photo that you see from the slide, the introductory slide, is exactly from that area. Okay. And if you have any questions, you should contact me. I can provide more details about that. Okay. But so yeah, that is our basis, and there is also even linguistic. The oldest presumed words for beans are from a group that is just in Guanajuato, so just east of Jalisco.
So Eric also has his hand up. I'll let him ask his question, and then you have a question, Dan, too.
Paul, first, thank you. A huge thank you for joining us, and thank you for making time for the students. I know several of them are really looking forward to this. Like Yolanda, I'm going to have to jump off after this, and my apologies for that. Somebody's committee meeting has taken far too long to happen. So the big question I have, I'm going to drop another question in the chat, is a long-standing one. What do you think the relative importance of standing variation versus new mutations is? And I can't help but ask it, but like I have to ask it just to scratch an itch. A more local question is, given that cranberry beans and kidney beans are out of the Andean gene pool, how do you think they got to the Northeast? There are indigenous groups here that claim cranberry beans, particularly the Jacob's cattle bean, as indigenous to the Passamaquoddy. Do you think they came with the French, or did they make an East Coast route through the Caribbean up here?
Yeah. Well, I would say there is a there's a record from a Native American woman who said distinguished between her beans and the beans of the white man. And it was the shape of the seed, and particularly the location of the hilum. So the hilum is that, the attachment of the seed to the placenta is within. And he said, if it's on top of the seed, these are my beans. The beans of the white man have the hilum sunk, it's sunk in the shape of the seed, which is like the kidney bean shape. And so my thing that the beans originally came up from what is now Mexico or Mesoamerica. They were the agriculture spread northwards. The same case for maize. And so there was, I think if you look at many of these indigenous beans, they were actually Mesoamerican. Now, I think the Andean beans were introduced probably mostly via Europe into North America. I'm sure that there are exceptions to that, but I would say that's the introduction. And so the two were meeting by different routes into in North America. Now you have to be careful that some of these materials have been bred, actually. A case is the Hidatsa yellow. Hidatsa is a Native American tribe from the central part of the country. What is being sold as Hidatsa yellow, or Arikara, there is one of them has actually been bred by the Canadians into a bush type. So you have to be careful about the names and not assume that if it has some trace or some link to Native Americans, they're not necessarily the original material. You have to be very careful about that. I think it's the Arikara yellow, I think, now that I think about it. Yeah. So I think that was the second question. The first question.
The first question was sort of a philosophical one across domestications about the relative importance of
de novo and standing variation. I think most of what we see is de novo mutation, like the determinacy. We didn't find any in wild beans. The P locus for white seeds, I don't think that this has been found in wild beans. It probably has very low fitness. Yeah.
Thank you, Paul. We have a question here from Dan. Dan, do you want to
Yeah, it might be a foolish question and there might not be a good answer, but like, why these 5 species of beans? How come all of the other ones have not been domesticated?
So could you repeat the question? Why 5 species of beans?
So Dan asked why these 5 species of beans and not any of the other wild species of beans? Like why were these ones in particular domesticated and not any of the other species that are currently wild in the Americas?
Yeah, I don't know. When you see, is it historical contingency? I think, I think your question should be placed in a more general kind of why do people adopt agriculture rather than continuing to do hunting and gathering, and which materials do they take? Is it just by chance, or do they have knowledge? There are a few other species that have larger seeds in Phaseolus. These are wild species, and people have wondered exactly the same question. Why weren't those species with larger seeds also domesticated? I think a possibility is that once you domesticate, you come back to the same materials. You don't keep domesticating new species. And so it's the similar questions. Why is common bean so much more important than, let's say, year bean, which has very limited distribution? Yeah. A colleague, Jim Beaver of Puerto Rico, asked me, so why is common bean so much more popular than lima bean, which is the second most important domesticated? I don't know.
Yeah.
So, but it's a really good question, and it's a question that currently people are asking themselves. And
Yeah.
it's also relevant in terms of adopting other domesticated species. See, if we are going to adopt peppery bean as a bean that is resistant to heat and drought, the biggest obstacle may be the preference for peppery for common bean rather than peppery bean. And the preference is built on the size of the seeds, the color of the seeds, the texture, the taste, and so on. You might consider these to be subjective traits, but people eat what they are used to.
Thank you.
Have people tried to domesticate the other ones, if even just for an experiment? Like, can you get domestic-like traits from the other species if you just continuously selected for that?
It's easier said than done. And that is something that you could say, well, what are the circumstances of the research that would lead to that? You have to get funding. This is probably more than 3 years. And even if you succeed to make it look like a domesticated bean, are people going to eat it or not? And so the question is very often, am I better off improving current species? And that has been the guiding thought of the industry. Say, let's continue to improve common bean rather than improving maybe peppery bean. There may be a change in that thinking, but it's not a trivial experiment to domesticate a species. And there's a lot of talk about that. It is called neo-domestication. Is domesticated a wild species, and people say, well, I can use genome editing to achieve this domestication syndrome. I think it's easier said than done.
Thank you, Paul. I want to be respectful of everyone's time. I have some questions for Paul, so I'm going to stick around and ask them if he has time. But otherwise, thank you. Thank you to those of you who stuck around, and you're welcome to ask more questions if Paul has time. If not, I'm gonna go right on ahead. I thought it was really interesting.
Hello?
Hello?
Yeah, I can hear you.
Can you hear me? Okay. There was like a loud noise and I couldn't hear for a minute. So I was interested in that the aspect of determinacy that it was selected for in the Andean population. And I'm wondering if that is a result in changes in cultivation, like cultivation practices. So for example, in Minnesota.
Yeah.
In the you have it binding up the plants, but in the Andes, is there a different cultivation practice that is more suited for determinate types or indeterminate?
Yeah, I think that you're getting there because We have speculated that it was when it was domesticated, when beans were domesticated, maize was not present yet, and that maize allows you to grow a pole bean. But if you look at the crops, the typically Andean crops are root crops. They maybe are a foot or 2 feet high at the most, and don't provide that physical support. So when you have a determinate mutation of the ones that we see, they really modify the plant into a bush type almost immediately in one step, actually, depending on the genetic background. So I think that that's probably what happened. Although I should say that in the Mesoamerican domestication, you have bush types, but they are indeterminate. So again, there is different ways of achieving the same thing, bush bean, whether it's determinate or indeterminate.
Right.
But the milpa, the milpa is widespread in Mexico and elsewhere. Eventually, that association between maize and beans spread to the Andes as well and elsewhere. Some of the best examples of a milpa are actually in Africa. Because what you see in Mexico, for example, is that it was traditionally Jalisco, you had milpa types, the milpa production. They introduced improved maize varieties that required more inputs. The seeds were more expensive, so the farmers concentrated on producing maize in monoculture, with providing fertilizers, using herbicides, and so on. So where do you have to produce? So the bean cultivation moved to the central part of the country, central and northern part. So from Guanajuato up to the north, Durango, Chihuahua, and so on. Zacatecas is a big state, bean producing state, and it became also a monoculture. Okay. Eventually, but that's fairly recent in the last decades. Yeah. And so the preferred model or type now is the so-called type 2, is a bush indeterminate, actually. The green bean, the preferred type, is a type 1, so a determinate bush, actually, for reasons of production and harvest, actually.
Jazz and Joseph, do either of you have questions you want to ask? Okay, I have one more, and then we can let you go. So at the beginning, you sort of talked about this transition in agriculture from us being recipients of wild crop species to being exploiters of these crop species. And I was wondering, this is maybe more of a philosophical question. Is all agriculture exploitation of the environment and resources, or do you think that there are agricultural systems that work with the environment and are more so manipulations and not exploitations?
Yeah. I think that the industrial agriculture is definitely exploitation. And people talk about sustainable intensification and agroecological kind of agriculture. It's still some form of exploitation. There's nothing we can do about it if we want to feed 7 billion or 9 billion people. The people think of agriculture, oh, that's natural and so on. Actually, I don't think that what you see even in the background is a natural vegetation. You don't see that in nature, one tree after the other in rows. And So if you have the opportunity to do a germplasm collection, maybe funded by the USDA, to a center of origin, for example, do it, because it is a fantastic experience to see agriculture, and especially wild vegetation, if it still exists, or at least modified wild, but it provides you with a basis of comparison, really. And the agriculture we see, is not natural, actually. Now I realize that we all have to be fed, and we are not going back. I mean, there is this, it's almost a reciprocal, it's a symbiosis. On one hand, we rely on these crops to feed ourselves and to clothe ourselves and so on. On the other hand, the crops, the fully domesticated ones, rely on us for their propagation, actually. They would not self-propagate, actually. So there is this, I think agricultural production has been able to follow population increase, but at what cost, actually? That's the issue, I think.
Right. Thank you. Before we let Paul go, since we've ran over a bit, are there any final questions, any parting thoughts? Just wanna leave space. Okay, if not, thank you so much, Paul, for sticking around and talking with us.
No, that's fine.
And
If you have
yeah,
any further question, you can contact me, and yeah.
thank you so much. We really appreciate it, and have an excellent weekend.
I will, and I hope you too.
Thanks. All right, bye.
Bye.